Alright, we're picking up with question 13 on the biology practice air test. This question is about bacterial resistance to antibiotics, and the question really is about the mutations themselves. So it says random mutations lead to this immunity, or this resistance. So which statement describes how this mutation occurs? So how do mutations happen? I've already selected A here. A is the right answer. Honestly, the other ones don't really look close. They're hard to even make sense of what they're trying to get you to think of with those. The thing that makes sense is A. The mutation occurs when nucleic acids, typically we're talking DNA, nucleic acid is DNA. Remember, DNA is deoxyribonucleic acid. DNA is a nucleic acid. Are replicated. We're talking during replication. When the DNA is being copied, that is the prime time for mutations to occur. And yeah, so A is the right answer. Next up. For this one, we see a plant cell. We see some labeled organelles for the plant cell, and the question below is asking to identify two cell structures involved in the function described. Place the structure number into each of the blank boxes. So again, you don't use every label. We're just labeling which numbers do we think will do this job. And this job in this case is modifying proteins and transporting them out of the cell. So we're looking at several things here. This is a chloroplast. That's related to making food. This is a mitochondrion. That's related to producing energy, using food to make energy. 5 is pointing to the nucleolus. We're inside the nucleus there. This is a vacuole, storage. That's for storage. Cell wall. That's for providing structure, strength to the membrane. And our remaining two are the ones that matter for us here. Number 6 is pointing to the endoplasmic reticulum. That is, again, typically for finalizing proteins, sort of like, again, like modifying them, that modifying step there. Part of that is for delivery, like how they're going to get shipped around to mostly inside the cell, but also potentially shipped out of the cell. And then the Golgi is number 2 up here, the Golgi body. There's one there and one here. The Golgi apparatus, or the Golgi body, its job is, again, for packaging, as far as it labels the protein with a little marker that identifies where it's to go. So if it's going to leave the cell, the Golgi kind of puts the address label on the protein, so it tells it where to go. So 2 and 6 are both related to modifying proteins and shipping them out. So 2 and 6, or 6 and 2, whatever. That would be our correct answers for there. Next. A scientist isolates a number of non-photosynthetic prokaryotes. Which structure would be found in these cells? Again, if you've watched the prior videos, you know we've talked a lot about prokaryotes and eukaryotes already. The answer here is what would we find in a prokaryotic cell? We would find cell walls. Prokaryotes have cell walls. That's bacteria. They have cell walls. Also, some eukaryotes do, right? Like plants have cell walls. We just saw that. Animal cells don't, but most other cells do, including prokaryotes, including bacteria. So cell walls, that's our answer. Chloroplast is an organelle. So is a Golgi apparatus. Those things we don't find in prokaryotes. Remember, they don't have membrane-bound organelles. And letter D is a nucleus. That's basically the other defining point. Prokaryotes do not have a nucleus. So B, C, and D are no good. Cell walls, A is the right answer. Alright, this one is about fermentation. Fermentation is a process that happens when there's not enough oxygen for cellular respiration to occur. Fermentation is going to help a cell to make a little bit of extra energy when it's past its limits. Usually we're talking about when we run out of oxygen and there's not enough oxygen to allow respiration to happen. So part A of this question is identify the products of fermentation by moving the product labels into the blank boxes. First of all, we're talking about, in the question it says how much CO2 is detected. CO2 is a product, like that's, so the more fermentation we get here, the more CO2 we're going to see. Fermentation produces CO2. That's where this is called alcoholic fermentation we're talking about. CO2 is a product. This is where the bubbles come from. That make bread rise, or that make cake rise. It's also where the bubbles come from that makes the holes in Swiss cheese. Those holes come from bubbles of CO2 that form inside the cheese during alcoholic fermentation. So the other product, since it's alcoholic fermentation, is ethanol. And this is where when people brew beer or wine, ethanol is the alcohol that forms, and they brew those things by fermenting. They ferment them, and so that's how they make their alcoholic products. The other type of fermentation is called lactic acid fermentation. Instead of making these two products, lactic acid fermentation makes lactic acid. That's the kind of fermentation that makes your muscles burn and that makes your muscles sore after a big workout. So there's two types of fermentation, but the one that we would commonly see is the one that makes CO2 is also the one that makes alcohol. That's alcoholic fermentation. So those are our products. The optimal fermentation temperature is the one that seems to produce the most CO2. Again, the more CO2 that gets made, the more products we must be producing. So the highest CO2 is here, and the highest CO2 for the second trial is here. So 35 degrees seems to be the best one. So we find 35, and that's our optimal temperature. So there's our 3 points for that one. Next. Many of you have seen this one already. I've shared this one in class. The evolutionary relationships among 4 animals and the traits that they develop along the way. Remember, these are derived characters. That means if you trace the history of evolution of these organisms, this is back in time, and as we branch off, we're looking at the evolutionary pathway. So for example, if we go this way, when we pass through this mark, these organisms have developed this trait. So they've developed vertebrae. Sharks split away and go in this direction in their history before we get to this point, which means sharks do not share that trait. So sharks have this trait, but not the one that comes after they split. Anybody who has to pass through one of these marks does have that trait. So, let's start with the shark. Sharks have vertebrae, and they do not have the other 3. The other 3 come after the shark splits away. Bullfrogs have vertebrae, and they also have 2 pairs of limbs. Kangaroos, vertebrae, 2 pairs of limbs and mammary glands. Vertebrae, 2 pairs of limbs, mammary glands. And last, the chimpanzee has all 4 of these traits. It's got vertebrae, it's got 2 pairs of limbs, it's got mammary glands, and it's got a placenta for baby's development. So those checkboxes hit the characteristics of each of the 4 animals in this cladogram. Which density-dependent factors, other than the predator-prey relationship, affected the populations of moose and the wolves on Isle Royale? Sometimes questions like this can be a little misleading. This graph actually has very little to do with answering this question. So this graph is here. You might see a graph like this one. This is kind of a famous study. Isle Royale is where it's a very isolated area, and so they've been able to study. Not a lot of things live there, but there are wolf packs and there are moose that live there, and they've studied for a long time the relationship between the moose and the wolf population. And so we see them traced out here, the wolf population in dotted lines, and that's down here, and then the moose population is a solid line. And what you can kind of see is, which isn't surprising, moose are hunted by the wolves. Wolves are predators of the moose. So what we see is sometimes when we see a swing in one species, like here we see the moose swing up, that goes right along with a drop in wolves at the same time, which shouldn't be a surprise. If there's less wolves, the moose population is going to be able to be a little bit bigger. And the same thing works the other way. We get a lot of wolves here. This is a big increase in wolves, and the moose population shrinks back down. And the shrinking in the wolf population, again, eventually that's going to make the moose population shrinking, means the wolf population will also drop again because there's less food. And so those two are very related to each other. You can kind of see the swings and kind of explain why they might be reacting to each other. But this question asks you about density-dependent factors. Density-dependent population limiting factors are things that cause the population to be limited in size based on how dense it is. So like the denser the population, the bigger this factor kicks in. So some of these things are not density dependent. The ones that most clearly are related to density are from letter D. For a moose, food availability, so again, this is a snowy, there's not a lot of food here, so food availability would be a density dependent limit for moose. The more moose there are, the bigger this becomes an issue. And for wolves, the same thing. The more wolves there are, the bigger of an issue disease can become. The more they're packed in together, the faster disease can spread. So that's letter D makes the most sense here. Again, some of these things are just not density dependent. Extreme temperatures, it doesn't matter if it's 1 moose or 10 moose or 100. If it's extreme temperatures, it could kill them, like, no matter what the population is. Same thing for flooding. It doesn't matter how crowded the wolves are, flooding would be deadly to any number of them. So D is the one that's the most density dependent. And we will stop there with this video and pick up with the next one.